السلام عليكم، معكم دانا أبو بكر من كلية طب الأسنان. إن شاء الله اليوم رح نكمل على الشابتر. أول شي رح نبلش بشرح the thymus. So the thymus is the production and exit site for T lymphocytes and it's part of the primary organs نفس ما حكينا قبل. The thymus is a gland and it has a bilobed structure in the mediastinum. Its main function is the induction of central tolerance, which along with the regulatory T cells prevents autoimmunity. رح نحكي عن هذا الموضوع أكتر in depth بعدين. The organ originates from the embryo's third pair of pharyngeal pouches اللي هم الendoderm, with precursor lymphoblasts circulating from the bone marrow to invade and proliferate in this unique thymic epithelium during its development. شو يعني the thymus is made of two important structures اللي هم أولاً the thymic endothelium, which is made of endoderm. بعدين عندنا precursor lymphoblasts, which are made of mesoderm, and they invade and proliferate the thymic epithelium. The thymus is fully formed and functional at birth, and the thymus remains large and very active in T cell production until puberty, during which it normally undergoes involution with decreasing lymphoid tissue mass and cellularity and reduced T cell output. So, a comparison between the thymus and the child and an adult. As you can see on the child, it's much larger than عند adult and it's well developed and fully functional. And عند الادult صار في involution. As you can see that it shrunk, and the size of the thymus gland in adults is نفس ما حكيت much smaller compared to children. هذا طبعا it happens once the child hit puberty. And نفس ما حكيت قبل إنه the cellularity decreases, the T cell output decreases, and the lymphoid tissue mass decreases. The thymus has a vascularized connective tissue capsule that extends septa into the parenchyma, dividing the organ into many incompletely separated lobules. So the thymus is separated by the septa into incompletely separated lobules. Each lobule has an outer darkly basophilic cortex surrounding a more lightly stained medulla. The staining differences reflect the much greater density of lymphoblasts and small lymphocytes in the cortex than the medulla. So the cortex has lymphoblasts that are denser and smaller lymphocytes than the medulla. As you can see here, هي السبتا dividing the thymus into lobules. As you can see, the cortex, the darkly stained part, and the medulla is the lightly stained part. Again, the septa, cortex, medulla again, septa, cortex, medulla, and so on. And this is the capsule. Okay? So, the thymic cortex contains an extensive population of T lymphoblasts or thymocytes, some newly arrived via venules located among numerous macrophages and associated with the unique thymic epithelial cells, اللي هم TECs, that have certain features of both epithelial and reticular cells. So عندهم both features of the epithelial and reticular cells. نفس مثلا الابيثيليال انه عندهم endoderm نفس ما حكينا قبل شوي, they're endodermal in origin, نفس lining of the epithelium. So شو يعني هاي؟ انه the thymic cortex, الكورتكس تبع الثايمس has a large population of T lymphoblasts. Some بيكونوا pre-T lymphoblasts and some بيكونوا mature T lymphoblasts. And some of them اللي pre بيكونوا they are newly arrived via the venules. And نفس ما حكيت إنه basically الكورتكس تبع الثايمس فيه a large population of T lymphoblasts. So these TECs have a large euchromatic nuclei, but are morphologically and functionally diverse. These are three major types of TECs in the cortex of the thymus. So هاي three types of TECs. أول type is squamous TECs, and stellate TECs, and the other squamous cortical TECs. رح نتكلم عنهم هلا in detail. So the squamous TECs, they form a layer joined by desmosomes and including junctions, which lines the connective tissue of the capsule and septa and surrounds the microvasculature. So the squamous TECs join مع desmosomes and occluding junctions, forming a layer which lines the connective tissue of the capsule and lines the septa as well as the microvasculature, which creates an isolated cortical compartment, and together with the vascular endothelial cells and parasites forms a blood-thymus barrier. So لما they form a layer, this layer joins with the vascular endothelial cells and parasites, forming a blood thymus barrier which prevents unregulated exposure of thymocytes to antigens. هلا هذا in early stages of development يعني pre-thymocytes, and this prevents the autoimmune reaction which the lymphatic system works greatly to prevent لأنه the autoimmune reaction is very dangerous to the body. Throughout this compartment, another population of stellate TECs with processes containing keratin tonofilaments joined by desmosomes form a cytoreticulum. So the cytoreticulum is keratin tonofilaments which are joined by desmosomes, and هذا الcytoreticulum macrophages and developing lymphocytes attach to the cytoreticulum instead of attaching to the reticulin fibers, and ما في reticulin fibers بالstellate TECs and in the thymus in general. Importantly, these cells are APCs, the antigen presenting cells اللي حكينا عنهم بالمحاضرة اللي فاتت, and they express MHC class 2 molecules in addition to class 1. Stellate TECs also secrete numerous cytokines for T cell development and other immune functions, justifying this organ's inclusion among endocrine glands. So, I believe there is a cross section, I think. Okay, هيها. As you can see هي الستالت TECs, هي large nuclei اللي حكينا عنهم, وهي cytoreticulum processes اللي بيكونوا موجودين عند الستالت TECs. As you can see هيهم. Okay, هي TEC اللي حكينا عنها الستالت TEC, and this is the large nuclei اللي بيكونوا عندهم. بيكون عند all of the TECs. Okay, and the last type is the other squamous cortical TECs, and they express MHC class 2, نفس الstellate TECs, but they form a sheet-like structure contributing to a functional cortico-medullary barrier between these two regions of each lobule. So, other squamous cortical TECs, they express MHC class 2 نفس الstellate TECs, and they form a sheet-like structure contributing to a functional cortico-medullary barrier between these two regions of each lobule. The more lightly stained thymic medulla contains fewer and larger, more mature lymphocytes. These three related types of medullary TECs form the following هلا رح نحكي عنهم. But to note إنه المدلة فيها fewer and larger and more mature lymphocytes than the cortex. هدول types of medullary TECs, they form the following هلا رح نحكي عنهم. Firstly, they form a second layer of boundary between the cortex and the medulla اللي هي cortico-medullary barrier. من اسمها cortico-medullary, it's a barrier between the cortex and the medulla. These are formed by medullary TECs. It also forms a cytoreticulum اللي حكينا عنه هلا, and هذا الcytoreticulum it supports the T lymphocytes, dendritic cells and macrophages, which are all less densely packed than in the cortex. It also expresses many specialized proteins specific to cells of other antigens, to other organs, اللي هم cell antigens. And بيصير في بيكون موجود عندنا large aggregates of TECs, sometimes concentration تبعهم concentrically arranged, called the Hassel Corpuscles رح نحكي عنهم هلا. And these Hassel Corpuscles, they have a diameter that's up to 100 micrometers, but they are variable in size. يعني مش كل ال Hassel Corpuscles بيكونوا the same size. And their cells secrete several cytokines that control activity of local dendritic cells, including factors promoting the development of regulatory T cells for peripheral tolerance. So شو يعني؟ الميدالري TECs form the لما يصير في عندهم large aggregates, they're called the Hassel Corpuscles, and these Hassel Corpuscles they have a diameter that's up to 100 micrometers, but as we said they vary in size, and they are only present in the medulla. يعني لما نشوف في عندنا Hassel Corpuscles نعرف إنه هاي المدلة. And these corpuscles secrete several cytokines that control the activity of local dendritic cells, and it also يعني high activity includes factors promoting development of regulatory T cells for the peripheral tolerance. The microvasculature of the medulla is not surrounded by a tight layer of TECs, and mature T lymphocytes exit the thymus by passing through the walls of venules and efferent lymphatics in this region. لأنه نفس ما حكينا الثايمس is the site of production and exit of T lymphocytes, فما في عنا anything that will prevent anything that wants to leave the thymus عشان the T lymphocytes can exit من الثايمس. اوكي هلا رح نتكلم عن the role of the thymus in T cell maturation and selection. This picture summarizes the whole process, so I'm going to explain on this picture, and everything that's said here, that's mentioned here will be mentioned لما أحكي عن هاي الصورة. أول شيء بدنا نعرفه إنه في the process of selection بالthymus is composed of two stages. أول شيء عنا positive selection stage and the negative selection stage. هلا positive selection stage بدنا نعرف عنه شغلتين. أول إشي takes place in the cortex, and positive selection سميت positive لأنه بدنا positive result منها, بدنا a yes result منها, which is the survival of cells depends on its ability to bind to MHC molecules. يعني if it binds to the MHC molecule, which is a positive result, خلاص it's gonna go to the next stage, which is the negative selection. If it doesn't, it's gonna die by apoptosis, and we're gonna talk about it now. The negative selection, however, happens in the medulla, and بدنا negative result منها, بدنا a no result منها, and the negative result اللي هي not recognizing self antigens, and we're gonna talk more about هدول selections in a moment. So أول شيء بدنا نحكي عن Thymic Cortex, the positive selection which takes place in the Thymic Cortex, وهذا بصير pre-T lymphocytes before the expression of protein and CD هو cluster of differentiation اللي حكينا عنه بالمحاضرة اللي فاتت. And هدول pre-T lymphocytes, if they bind to MHC molecules, then they go to the next stage, which is the negative selection. If they don't, they're gonna die by apoptosis because they're gonna be deemed as non-functional lymphocytes. So هدول pre-T lymphocytes will be exposed to the MHC molecules, and if they bind, they are going to نفس ما حكينا they're going to survive and advance to the medulla, and if they don't, they're gonna die by apoptosis. هلا بالnegative selection which takes place in the thymic medulla, negative selection نفس ما حكينا survival of the T lymphocytes will depend on its inability to recognize the self antigens. So if it doesn't recognize the self antigens, then it'll survive. If it does, it'll die by apoptosis. So هاي ال pre-T lymphocyte, هلا negative selection أول شيء it tests the TCR, the receptors, the T cell receptors. هدول T cell receptors, if they recognize نفس ما حكينا the MHC molecules, the self antigens اللي بيكونوا موجودين على surface of the MHC molecules, if they do recognize them, they're going to die by apoptosis. If they don't, then they're gonna survive. هلا هاي as you can see, هاي ال MHC class 1 and class 2 for example, يعني on their surface هدول the blue circles almost هدول self antigens. The TCR should not recognize the self antigens. And again, I'm going to repeat, if it does not recognize the self antigens, it will survive and it will complete the maturation. If it does, it will die by apoptosis. هلا 2% of the cells survive the selection process in the thymus, and this is because the remaining 98% are either non-functional or self-reactive. If the T cell receptor recognizes the self antigen, بنحكي إنه هاي it's a self-reactive cell, وإحنا ما بدنا any self-reactive cell in our body لأنه self-reactive cells will cause autoimmunity, so they have to die by apoptosis. That's why they're killed. That's why they die. They خلص يعني. But if they don't, نفس ما حكينا, they're gonna survive. So the remaining 98% من هدول 100% of the T lymphocytes, 2% they survive. The remaining 98% are non-functional and self-reactive, وهاي self-reactive cells, their presence lead to autoimmunity. بعدين بعد ما يصير في عنا complete maturation, مش complete maturation, and the maturation will be completed now لما يصير في عنا loss of the cluster of differentiation. لما يصير في عنا هدول 2% they pass and they survive the selection, بيصير في عنا إنه they have to pick whether they want to lose their CD8 or lose their CD4. If they choose to lose their CD4, بيصير في عنا cytotoxic T lymphocytes, and this cytotoxic T lymphocyte only retains the CD8 protein, losing the CD4. And if they choose to lose the CD8, بيصيروا helper T lymphocytes, and نفس ما حكينا they only keep their CD4 protein. فإذا عندهم CD4 بيصيروا helper T lymphocyte, إذا عندهم CD8 بيصيروا cytotoxic T lymphocyte. And لما they select whether they want to lose the CD4 or CD8, they complete the maturation وخلاص they get released من الثايمس. And this is the end of the thymus. هلا رح نتكلم عن mucosa-associated lymphoid tissue. So the mucosa-associated lymphoid tissue. The mucosa-associated lymphoid tissue, also known as the MALT, is a part of the secondary lymphoid structures, the secondary lymphoid organs, along with the lymph nodes and the spleen. The mucosa or inner lining of the digestive, respiratory, and genitourinary tract is a common site for invasion by pathogens because their lumens are open to the external environment. So, for example, pathogens can enter the digestive tract through the food that we ingest, respiratory through the air that we inhale, genitourinary through sexual intercourse, for example. And they can enter easily because they are entrance sites since their lumens are open to the external environment. To protect against such invaders, mucosal connective tissue of these tracts contain large and diffuse collections of lymphocytes, IgA-secreting plasma cells, APCs, lymphoid nodules, all of which comprise the MALT. So the MALT is comprised of lymphocytes, IgA-secreting plasma cells, APCs, and lymphoid nodules. Lymphocytes are also present within the epithelial lining of these tracts, and most of the immune cells in MALT are dispersed diffusely in the connective tissue. Others are found in aggregates forming large conspicuous structures such as the tonsils, Peyer patches, and the appendix. رح نحكي عنهم هلا. Collectively, the MALT is one of the largest lymphoid organs. The largest lymphoid organ is the spleen. MALT is one of the largest, and it contains up to 70% of all the body's immune cells. Most of the lymphocytes here are B cells. Among T cells, CD4 helper T cells predominate. So most of the lymphocytes اللي موجودة بالمالت are B cells. في عنا T cells, but these T cells are mostly CD4 helper T cells to help those B cells. Tonsils are large, irregular masses of lymphoid tissue in the mucosa of the posterior oral cavity and nasopharynx, where their cells encounter antigens entering the mouth and the nose. Named by their location, these masses are the palatine, lingual, and pharyngeal tonsils. Something to note is that tonsils are not fully encapsulated, and in all tonsils, the lymphoid tissue is closely associated with the surface epithelium. So the epithelium is the same as the epithelium of their location. For example, the ones in the respiratory tract have a columnar epithelium, I believe. Yes, a ciliated columnar epithelium. We're going to talk about it in details now. So the palatine tonsils, the first tonsils we're going to talk about are the palatine tonsils. They're named palatine because they are located posteriorly to the soft palate, and they are covered by stratified squamous epithelium. The surface area of each is enlarged with 10 to 20 deep invaginations, epithelial invaginations, which are also called the tonsillar crypts, in which the epithelial lining is densely infiltrated with lymphocytes and other leukocytes. The lymphoid tissue is filled diffusely with lymphocytes, with many secondary lymphoid nodules around the crypts. This tissue is underlain by dense connective tissue acting as a partial capsule. The second type is the lingual tonsils. Lingual and palatine tonsils have many structures that are similar. The lingual tonsils are situated along the base of the tongue. They are covered by stratified squamous epithelium with crypts and have many of the same features as palatine tonsils, but they lack distinct capsules. The capsule in the palatine tonsils you can see them clearly, whereas in the lingual tonsils they are ill-defined. The single pharyngeal tonsil is situated in the posterior wall of the nasopharynx, and it's covered by pseudostratified ciliated columnar epithelium and has a thin underlying capsule. The mucosa with diffuse lymphoid tissue and lymphoid nodules is invaginated with shallow enfoldings but lacks crypts. So بالpharyngeal tonsils ما في عنا crypts. And the fourth type, which is not mentioned in the book but the professor has mentioned it, is the tubal tonsils. The tubal tonsils are they surround the openings of the eustachian tubes. The eustachian tubes are tubes that connect the throat to the middle ear. They open into the nasopharynx, and ما عندهم a capsule. They also contain elastic fibers. So هاي الصورة أخذتها اليوم المحاضرة. We're going to talk about it هلا, and it's a comparison of the tonsils that we just mentioned. I can't use the pointer. I can, okay. So I thought I couldn't. I can. Anyway, the palatine tonsils are the palatine, the pharyngeal, and the lingual, and the tubal tonsils, they all have lymphoid nodules which are sub-epithelial in position. They have lymph vessels, obviously, but there are no efferent vessels. They are all efferent. Blood vessels here are the arteries that enter through the attached surface. The capsule is prominent in the palatine, less definite, ill-defined in the pharyngeal. In the lingual, it's either thin or absent, and there is no capsule in the tubal tonsils. The cortex, medulla, and the hilum are absent in all of the types of the tonsils. The epithelium in the palatine and lingual are stratified squamous, whereas in the pharyngeal and tubal, they are pseudostratified columnar epithelium, نفس the respiratory tract. And features to differentiate between them: the palatine, they have 10 to 20 crypts. The pharyngeal, no crypts at all. The lingual has 1 crypt, and the tubal is associated with the Eustachian tube. So, let's talk about this medical application, which is the tonsillitis. The inflammation of the tonsils, التهاب اللوز, and it's more common in children than adults. ليش? Because in children, as we said, children will gain acquired adaptive immunity as they get exposed to different things. So at first ما بيكون عندهم immunity to such illnesses. So they are prone to getting tonsillitis, and they get it commonly. بعدين as time goes and as they get exposed to it, بيصيروا immune to it, and it will happen rarely, but it still does happen. So chronic inflammation of the pharyngeal, lymphoid tissue, and tonsils of children often produces a hyperplasia and enlargement of the tonsils to form adenoids, اللي هي اللحمية. And هاي اللحمية, or the adenoids, are located in the upper part of the posterior wall of the nasopharynx, and on the sides of the adenoids في عنا openings of the Eustachian tubes. And the enlargement of tonsils can obstruct the Eustachian tubes and lead to middle ear infections, and this middle ear infection is caused by accumulation of mucus, which will lead to hearing loss. So symptoms of tonsillitis in children is the inability to swallow food, inability to breathe, snoring, sleep apnea, and so on. So the tonsillitis, لما التونسلز لما اللوز بيكونوا ملتهبين وبيكونوا enlarged, بيكون عنا a viral infection, and لما يكونوا ملتهبين وكبار وعليهم white spots بتكون a bacterial infection. In the case of a bacterial infection, the doctors will prescribe an antibiotic. In a viral infection, of course, they won't prescribe an antibiotic because antibiotics do not work on viruses. However, if it was a viral infection and doctors found that the immunity is low, they will prescribe antibiotics to increase the immunity. So, diffuse MALT that extends from the pharynx along the entire gastrointestinal tract becomes very well developed again in the mucosa and submucosa of the ileum. And here large aggregates of lymphoid nodules comprise the Peyer patches. So the Peyer patches are present in the ileum, and as نفس tonsils, they are large aggregates of lymphoid nodules, each containing dozens of nodules with no underlying connective tissue capsules. So the Peyer patches are present in the ileum, عندهم dozens of nodules, ما في عندهم capsules. The simple columnar epithelium that covers the lymphoid nodules of Peyer patches includes scattered large epithelial M cells with apical microfolds. So on their apical surface عندهم microfolds, ما في عندهم a brush border. If you remember, brush border the densely packed microvilli that's covered with glycocalyx and to increase the surface area. In the M cells ما في عليهم brush border, عندهم apical microfolds. And these M cells are a unique epithelial cell type specialized for the uptake of particles and intact microorganisms. The basolateral surface of the M cells have a deep invagination, which forms a large pocket open to the underlying lymphoid tissue through a unique porous or sieve-like basement membrane. So the pocket high is open to the underlying lymphoid tissue اللي بيكون تحت الأم cells through a uniquely porous or sieve-like basement membrane, sieve يعني مصفاية, and containing a transient population of lymphocytes and dendritic cells. Antigens in the intestinal lumen are continuously sampled at the apical surface of these cells and transferred to the immune cells in the pockets. So on the apical surface of the M cells في عنا microfolds وفي عنا continuous sampling. On the basolateral surface في عنا invaginations, deep invaginations forming pockets which are open to the underlying lymphoid tissue through a porous basement membrane. Lymphocytes and dendritic cells leaving the M cell pockets through the basement membrane pores interact and initiate adaptive responses to the antigens, which result in the formation of the secondary lymphoid nodules. Locally produced B cells give rise to the plasma secreting cells اللي هم ال IgA. Locally produced B cells give rise to the plasma cells secreting IgA. The plasma cells are the ones that secrete IgA, which is transported by enterocytes into the intestinal lumen to bind and neutralize the potentially harmful agents. Another significant collection of MALT occurs in the mucosa of the appendix. So احنا حكينا هلا عن two MALT aggregates. أول شيء ال tonsils، حكينا كمان عن ال Peyer patches. هلا رح نحكي عن appendix. So the appendix is a short, small diameter projection from the cecum. حكينا عنه بال GI tract if you remember. And typically the mucosa of the appendix is almost completely filled with lymphoid tissue, effacing the glands otherwise found in the large intestine wall. The lumen contains the normal bacterial flora of the large intestine and may serve to retain some of these beneficial bacteria there during diarrheal and illness. So the appendix is a short, small diameter projection from the cecum. The mucosa of the appendix is filled with lymphoid tissue, which efface the glands otherwise found in the large intestinal wall. The lumen of the appendix contains normal bacterial flora, and this may serve to retain. The bacterial flora بيكون موجود بالlarge intestine and the appendix, and the appendix will serve to retain some of these beneficial bacteria. مش هم إنه it's a normal bacterial flora, so the appendix will retain some of the bacteria which can be beneficial during diarrhea. Moving on to the lymph nodes. So the lymph nodes are bean shaped. الدكتور حكى إنه they're not actually bean shaped بس ما حكى what shape they actually are. He said I think إنه they differ in shape. However الكتاب مكتوب bean shaped so رح نعتمد bean shaped. They are encapsulated structures, generally only 10 mm by 2.5 cm inside, and they are distributed throughout the body along the lymphatic vessels. نفس ما شفنا if you remember شفنا بأول صورة in this chapter. The lymph nodes scattered throughout the body, but they are gathered specifically in certain areas, for example عند the armpits and the neck and the ears, and they are scattered throughout the whole body, but they are present in groups in certain areas. So a total of 400 to 450 lymph nodes are present in the axillae, armpits, and the groin, along the major vessels of the neck and in the thorax and abdomen, and especially in the visceral mesenteries. The nodes constitute a series of inline filters of lymph that defend against the spread of microorganisms and tumor cells and provide enclosed environments for antigen presentation and development of plasma cells secreting non-IgA antibodies. So the function of lymph nodes, أول إشي, their main function is to filter. They filter the lymph coming from the circulation. They provide an enclosed environment for antigen presentation and development of plasma cells secreting non-IgA antibodies. Before merging with the bloodstream, all lymph is filtered and has antibodies added by at least one lymph node. So before ما يكمل lymph mass circulation, all the lymph is filtered and the antibodies are added to the lymph by the lymph nodes. So another function of lymph nodes is to add antibodies to lymph. Embedded in loose connective tissue, a lymph node has a convex surface where afferent lymphatics enter and a concave depression, the hilum, it's pronounced hilum, where an efferent lymphatic leaves and where an artery, vein, and nerve penetrate the organ. رح نشوف هلا بالصورة تحت. A dense connective tissue capsule surrounds the lymph node. So the lymph node is embedded in loose connective tissue, surrounded with a dense connective tissue capsule. عندها كمان extending trabeculae internally through which the blood vessels branch. Valves in the lymphatics ensure unidirectional lymph flow. The most abundant cells of the lymph nodes are lymphocytes, obviously. So the lymph أكتر نوع من أنواع السيلز موجودة فيه الليمفوسايتس of all types. عندهم كمان plasma cells، عندهم dendritic cells، عندهم macrophages and other APCs. وعندهم كمان FDCs. FDCs اللي هم الfollicular dendritic cells. في كمان FDCs بالlymphoid nodules. All of these are arranged in a stroma of reticulin fibers and reticular cells to form three major regions with each lymph node. The three major regions are the cortex, the pericortex, and the medulla. The cortex and the pericortex, separating them is the pericortex. You can't really tell where the pericortex is, but there is one thing that differentiates. هلا رح نعرف إيش هو as we go. So the cortex contains the nodules, whereas the pericortex does not contain nodules. So that's how you differentiate them. In a cross section, for example, شفنا nodules بنعرف إنه this is the cortex. ما لقينا nodules رح نعرف إنه إحنا بالpericortex. لقينا medullary sinus and medullary cords رح نعرف إنه إحنا بالmedulla. So the outer cortex, the outer part, contains nodules. Pericortex does not contain nodules. And the pericortex is the deeper extension of the cortex. وعندنا finally the medulla with a prominent draining sinusoid that's adjacent to the hilum. Unlike the thymus, these regions of lymph nodes are not compartmentalized by the epithelium, and the cortex includes the following components. So the cortex is made up of the subcapsular sinus, which is immediately inside the capsule, and it receives lymph from the efferent lymphatics. From this space, cortical sinuses or trabecular sinuses branch internally along the lymphoid nodules along the trabeculae. These sinuses, اللي هم cortical sinuses, are lined by a very thin discontinuous endothelium penetrated by reticulin fibers and processes of dendritic cells. So the cortical sinuses are lined by a very thin discontinuous epithelium. يعني بيكون في holes في fenestries بالendothelium penetrated by reticulin fibers and processes of dendritic cells. Lymph containing antigens, lymphocytes, and APCs passes through these sinuses and percolates easily into the surrounding lymphoid tissue. Okay? So, the second structure اللي موجود بالcortex is the lymphoid nodules with or without germinal centers. يعني some في عندهم germinal centers and some don't. These lymphoid nodules consist largely of developing B lymphocytes and occupy much of the cortex not filled with helper T lymphocytes. So, إذا الكورتكس ما كان في إذا مثلا بهذا المكان ما كان في T lymphocytes أكيد في عنا developing B lymphocytes. Each nodule is organized around the long interdigitating process of FDCs, but these are not readily seen by routine light microscopy. Numerous macrophages are also present for removal of newly formed defective B cells that undergo apoptosis. The region between the cortex and the medulla is called the paracortex. It separates the cortex and the medulla. نفس ما حكينا the pericortex is an extension من الcortex, and it does not have precise boundaries but can be distinguished from the outer cortex by its lack of nodules. Unlike the superficial cortex, the pericortex contains lymphoid tissue that's rich in T cells, distinguishable by the immunohistochemistry. Also located mainly in the pericortex, specialized post-capillary venules called the high endothelial venules, the HEVs اللي هم high endothelial venules موجودين بالpericortex, and these HEVs represent an important entry point for most circulating lymphocytes into lymph nodes. So البericortex في ما في nodules بس موجود فيه عنا lymphoid tissue that's rich in T cells, وكمان ال high endothelial venule which is important اسمهم كمان HEVs, which is an important entry point for most circulating lymphocytes into lymph nodes. 90% of the circulating lymphocytes بالlymph nodes enter through the HEVs اللي موجودة بالpericortex. Endothelial cells of these vessels become unusually enlarged or cuboidal and express specific apical surface glycoproteins that mediate tethering and apodices of B and T cells from the blood into the pericortex of the lymph node. HEVs also occur in the large accumulations of MALT discussed previously, but are less well characterized in those tissues. So في عنا HEVs in two areas in the body. أولاً بالpericortex كمان موجودين عنا بالMALT, but in the MALT they are less well characterized. So the medulla, which is the last structure in the lymph node, has two major components, اللي هما medullary cords and medullary sinuses, and these how we differentiate, we distinguish between the medulla, the cortex, and the pericortex. So the medullary cords are branched cord-like masses, نفس الأسلاك, of lymphoid tissue extending from the pericortex. So The medullary cortex extends from the paracortex and they are branched or cord-like masses of lymphoid tissue. They contain T and B lymphocytes and many plasma cells. The medullary sinus, which is the second structure that distinguishes the medulla, are dilated spaces lined by discontinuous endothelium that separates the medullary cords. The lumens of medullary sinuses include a meshwork of processes from the reticular cells, which represent a final lymph filter. These sinuses contain many macrophages and sometimes neutrophils if the lymph node is draining an infected regions. They are continuous with the cortical sinuses and converge at the hilum as the efferent lymphatic vessel. So, quick recap, المدلة two major components. أول شيء عن medullary cords which extend from the paracortex. They are branch-like cord masses of lymphoid tissue and they contain T and B lymphocytes and plasma cells. Medullary sinuses are dilated spaces lined by discontinuous endothelium, separates the medullary cords. The lumen of medullary sinuses include a meshwork of processes from the reticular cells, and they represent a final lymph filter before the lymph leaves the lymph node. These sinuses contain many macrophages and sometimes neutrophils. بيكون في موجود neutrophils بس لما تكون the lymph node is draining an infected region. They are continuous with the cortical sinuses and converge at the hilum as the efferent lymphatic vessel. So the medullary sinuses بيصيروا بعدين the efferent lymphatic vessel. So, okay. This is the lymph node. As you can see here, this is the structure. This is the B shape. هي عنا concave depression here. It has a convex surface. بس في عنا concave depression مكان الهايلم. So as you can see here عنا الافferent lymphatics اللي بتكون موجودة على the surface of the convex outer surface, and it's the entrance site for lymph. Directly it comes into contact with the subcapsular sinus اللي حكينا عنهم, and بعد ما تدخل على الليمف بعد ما الليمف enters ويصير in contact مع subcapsular sinus, it passes to the trabecular sinuses and then the medullary sinuses, and then it leaves via the efferent lymphatics. So as you can see here, هي this is the cortex, this is the lymphatic nodule, one lymph node, and بعدين the germinal center. Okay, some of them might not have a germinal center, or the purple part is the mantle zone. بعدين عندنا المدلة هي medullary sinuses as you can see, and هاي medullary cords نفس الأسلاك. So the medullary cord separates it acts like يعني هيك بين medullary sinuses and it separates them. بعدين عندنا paracortex, as you can see ما في عندهم ملامح, يعني there is no way to differentiate. The cortex again, the medulla and the paracortex, which is in between, as you can see, the cortex. So هاي الcortex كله, هاي المدلة, and in between them في عنا paracortex. The medulla is close to the hilum and the cortex is directly below the connective tissue capsule. So بعدين عنا okay, هاي as you can see هاي الafferent lymphatic vessels where the lymph enters. عنا طبعا valves هون to avoid the backflow. Here's the efferent lymphatic vessel where the lymph leaves after it's filtered, it leaves to the circulation. The concave surface مكان الهايلم, and it's also a site, as you can see here, it's a site for exit of efferent vessels نفس ما حكينا. It's a site of entrance of arterial supply here through here عشان يروحوا, it supplies the cortex and the lymph node in general, and it's a site of exit for the venous drainage. Okay, so the role of lymph nodes in the immune response. The lymph arriving at the lymph node contains antigens free in solution or bound to antibodies or complement, still on microorganisms or already internalized and transported by APCs. So the lymph that arrives to the lymph node بيكون in different situations: antigen free in solution, bound to antibodies or complement, still on microorganisms, already internalized and transported by APCs. If draining from an infected or inflamed region, lymph may also contain microorganisms or cytokines. So لو لقينا cytokines for example بالlymph رح نعرف إنه the lymph node is draining an infected region. Antigens not yet phagocytosed will be internalized by APCs in the lymph nodes and presented on MHC class 2 molecules. Circulating B and T lymphocytes traffic from node to node, يعني they circulate the lymph nodes and they go from node to node, and they enter the lymph node via the lymph or the HEVs اللي حكينا عنهم هلا اللي موجودين بالباراكورتكس, where B cells contact antigens on FDCs, follicular dendritic cells, and T cells sample antigens presented on dendritic and other APCs. So the B and T lymphocytes circulate the lymph nodes and they traffic from node to node, يعني they move from node to node. They enter the nodes via the lymph or HEVs, and once they enter, the B cells contact the FDCs, and the T cells sample antigens presented on dendritic cells and other APCs. The lymphocytes whose receptors recognize such antigens will be activated. So the activation of lymphocytes is when they recognize antigens, and the B cells will proliferate rapidly in germinal centers of follicles, cortex, with the help of the TH cells, often enlarging the entire lymph node. So B lymphocytes will proliferate rapidly with the help of the TH cells, which will result in the enlargement of the lymph node. Activated cytotoxic T cells in the paracortex proliferate to a much lesser extent without forming follicles. So the T cells will proliferate as well, but not as rapidly as the B lymphocytes. Many newly made B cells now activated against a specific antigen differentiate as plasma cells and move to the medulla or to downstream sites beyond the lymph node, where they produce antibodies. So the newly made B cells, which are immune to the certain antigen, will produce antibodies, and the specific TH cells or the CTLs or the Tregs اللي حكينا عنهم بالمحاضرة اللي فاتت also recirculate in the efferent lymph, and with the antibodies spread the immune defenses against those microorganisms throughout the body. So the TH cells, the CTLs, the Tregs, and the B cells will spread immune defenses against the pathogens, against the antigen throughout the body. Both B and T memory cells also move elsewhere in the body, providing long-lived protection and proliferating more rapidly upon subsequent exposure to their specific antigen. So the B and T memory cells that اللي بيصيروا بعدين memory cells also will move elsewhere in the body through the bloodstream, and they will provide a long-lived protection and proliferate more rapidly upon subsequent exposure to their specific antigen. So just one last thing, هاي a section of a lymph node. As you can see, هاي connective tissue capsule, هاي cortex, عندنا هون paracortex. As you can see ما في أي شيء هون that will distinguish إنه هاي paracortex. As you can see it's empty. هون في عنا nodules. Okay, هون في عنا sinuses and cords. Whereas هون ما في إشي that will يعني distinguish paracortex من cortex and medulla, for example. بس إنه it's in between them. ما فيها إشي. It's empty as we said. وهي trabeculae, which are like processes which are throughout the lymph node. So بهيك نكون خلصنا محاضرة اليوم. إن شاء الله المحاضرة الجاية رح نتكلم عن الspleen, and we're gonna end the chapter. For any questions, as always, feel free to message me on Messenger, and thank you for watching.